Yes, lads. Alright, this is our second MCBHD lecture, Patterns of Inheritance. Okay, we'll start Bismillah. So we start off with the family tree, aka the pedigree. So the family tree helps establish the family agenda and dynamics. And then gender and dynamics basically means how each person is related to the other. So it helps also narrow down the diagnosis, and it helps target and interpret genetic investigations. Cool, we move on. So these are the basic pedigree drawings. Males are squares, females are circles, partners have a line between them, siblings have a line above them, the line down is for children, and the affected ones are shaded, and the carrier ones have dots. Cool, easy peasy. Let's carry on. So there are also lots of other features as well, and this one I'll bring you close up. So as you would start from the top, across through it means deceased. We go to the left, two lines between a partner means a consanguineous marriage, which means like an interfamily marriage. As you can see here, it's between two cousins, because these two brothers are related. These two brothers are related, and the daughter and the son of those two brothers are married. So those two are cousins. So that's a consanguineous marriage. The ones in red are a stillborn baby of unknown sex with a diamond shape with an SB next to it. A triangle like that means spontaneous abortion, and a triangle with a line through it means therapeutic abortion. We then go on to the greenish ones, but for some reason they look blue. That means, as we can see, the triangle, the kind of like triangle with the two lines, this kind of shape. I have no idea how to explain it. The pyramid, I guess. It means identical twins, and just two lines between them means non-identical twins. The period in the diamond means pregnant, and the unaffected person whose sex is unknown is just a single diamond, and no offspring is those two lines. Cool. Let's carry on. So, focus is terrible. When we start a pedigree, we start with the individual we're talking to, and usually draw an arrow to them. So, for example, if a patient was to come into the hospital, and you to identify or you to investigate that person and that person's family, you would draw him, and you draw an arrow towards him. So you draw an arrow to that person, and I'm going to show you that here. So there's the arrow. So imagine this was our patient. We wanted to investigate him and why he's affected, and like what's going on with his family and the type of disease it might be. Then we'll draw an arrow to him. Cool. So the oldest siblings are on the left and the youngest siblings are on the right, and we try to put the same generations on the same line. Easy peasy. So we start off with the first type of kind of genetic disease, which is autosomal dominant. So we go on to autosomal dominant, aka AD just for short. Okay, so where there's an alteration in one copy of the gene or allele, and we only need one faulty allele for the disease. It manifests in heterozygous form, e.g. capital A and A, not two small a's, so you only need one faulty allele, and this faulty allele has to be dominant, obviously, and that's represented by a capital, and not whereas recessive is represented by a lowercase letter. So dominant represented by capital letters, recessive is I can't remember the word I just said. Recessive is represented, that's the word, recessive is represented by a small lowercase letter. So, genetic alteration may cause disease by an abnormal protein, making the gene disease-causing, or not having enough normal protein. So, the genetic alterations are basically caused by an abnormal protein being made, or not having enough normal proteins. So, autosomal dominant disorder is multiple Autosomal dominant disorders cause multiple generations to be affected. If they're affected, there's a 50% chance of passing the allele on, even if you're male or female, and therefore there's a 50% chance of your child being affected. Cool. So we'll go on to the Punnett square. So an example of an autosomal dominant disease is Huntington's disease. The capital H is autosomal dominant, the lowercase h is autosomal recessive. As you can see through the Punnett square, the being affected is 50% chance, whereas being not affected is also 50% chance. An important point is an autosome is any chromosome that is not a sex chromosome. So, an autosome is any chromosome that is not a sex chromosome. Cool. So characteristics of inheritance patterns in autosomal dominant diseases. So most individuals have an affected parent, but not everyone, because it may be a case of a new mutation. And secondly, there may be incomplete penetrance, where a parent may have the genotype for autosomal dominant disease, but may not express the phenotype. If you didn't understand that, relax. I'll go through it again. So the most individuals with an autosomal dominant disease, autosomal dominant disease, tongue twister, have an affected parent. Most individuals have an affected parent, but not everyone does, because number one, this individual may have a new mutation which may cause the disease, and number two, there may be incomplete penetrance, where the parent may have the genotype for autosomal dominant disease but may not express the phenotype, and we'll come on to more about penetrance later on. Carrying on, males and females are equally likely to inherit the alleles. However, there is a risk for each child being affected, which is 50%. We'll quickly look at autosomal dominant pedigrees. So here we can see 3 pedigrees for autosomal dominant. We'll start off with the one on the left. So here we can see autosomal dominant pedigree, as the disorder is present in each generation, and about 50% of each generation is affected. Easy peasy. So autosomal dominant disease, it's present in each generation, and about 50% of each generation is affected. As you can see, in the first generation, there's 3 people affected, and in the second generation, there is 5, so it's quite close. We'll then look at another pedigree, and this is a pedigree with autosomal dominance being 100% penetrance. However, this is an autosomal dominant pedigree with 67% penetrance, so 2 out of 3 penetrance. And why this one is 100% penetrance is that all the people with the genotype express the phenotype, and how we know all the people with the genotype express the phenotype is that basically whenever the parent has the disease, there is likely a child to have the disease. There is no child that has the disease without their parent having the disease. There is no child who has the disease without their parent having the disease. So for example, this parent has the disease, therefore this child has the disease. If this child had the disease and this parent, ignore that. I'm gonna show you. Basically, this guy has the disease. This guy doesn't. This guy has the disease. If this guy had the disease, then this guy would have to have the disease. Basically, that is if penetrance was 100%. I think I'm making it more complicated than I am, so I'm just gonna read the script. The left is 100% penetrance, so everyone who carries the disorder or the genotype will express it. So in this pedigree, everyone with the genotype expresses the phenotype. فقط. However, in this pedigree, if the penetrance is less, you would see less affected individuals, and unaffected individuals, and unaffected individuals goes on to having an affected child. So I'm going to repeat that. If penetrance is less, you would see less affected individuals. So as we see in this one, there's less affected individuals. Clear, there's less affected individuals in this pedigree than in this pedigree. And secondly, an unaffected individual goes on to having an affected child. So here's a perfect example. Unaffected individual has an affected child. However, over here, when there's an affected individual, he has an affected child. There is no child who in this pedigree that is affected without their parent being affected. There is no child in this pedigree that is affected by the disease without their parent being affected. And you're probably thinking, alright, what about this? Basically, these kids, they just got lucky and they didn't get the disease. But you don't see this. This is so important. Here, this child is affected by the disease, but somehow their parent isn't affected by the disease. That's because the parent has incomplete penetrance, as we said before. They have the genotype, but they're not expressing the phenotype. So we'll go over this very quickly again. The left is 100% penetrance, so everyone who carries the disorder or the genotype will express it. Number 2, this one, the penetrance is less. So if the penetrance is less, or because in this one the penetrance is less. So if the penetrance is less, you would see less affected individuals. So there's less people affected, and an unaffected individual goes on to having an affected child. An unaffected individual right here goes on to having an affected child. An unaffected individual goes on to having an affected child. Simple. Wow. Other aspects of autosomal dominant inheritance. Bloody hell, not penetrance again. So as you were saying, penetrance, the proportion of individuals with the genotype that express the phenotype. 95% penetrance would mean that 95% with the genotype express the disease phenotype. So 95% penetrance would mean 95% of people with the genotype would express the disease phenotype. Expressivity, so it's a variation of the severity of the disorder. So not everyone with the phenotype for polydactyly will express it to the same degree. So some may have tiny extra flap on the skin, whereas others may have a fully functioning extra digit, and polydactyly means an extra finger. So expressivity is the variation in the severity of the disorder. So some people express the disorder severely, having an extra finger. Some people express the disorder very minutely by having an extra flap of skin. Cool? So we move on to new mutation rate. This is the rate at which de novo mutation occurs. New or de novo mutation is when autosomal disease occurring for the first time in a family tree. So a new or de novo mutation is where there's an autosomal disease occurring for the first time in a family tree. Therefore, this autosomal disease will not be seen in previous generations. This rate varies between autosomal disease conditions. So the rate of de novo or new mutations varies between AD conditions. Reproductive fitness. So in some AD conditions, the affected are unable to reproduce. Therefore, the disorder is maintained in the population by new or de novo mutations. Simple. Germline mosaicism, what a word, or gonadal mosaicism. Wow, what words, man! This is madness. They are a new mutation arising during oogenesis, or spermatogenesis, and those are the development of the ovaries or the sperm. So, germline mosaicism is a new mutation occurring during oogenesis or spermatogenesis in the parent. So the parent isn't affected, but the child is affected, unfortunately. We'll then go on to parental age effect. So the chance of a new mutation increases with advancing parental age. Again, the chance of a new mutation increases with advancing parental age. And finally, anticipation. So this is the worsening of the disorder as you go down the subsequent generations. For example, offspring begin to be affected younger and younger. So, anticipation, offspring begin to be affected younger and younger. So, for one generation, the effect or the disease may start happening at 20 years old, and then the next generation might start happening at 15 years old, and the next generation, the disease might start happening at 10 years old. You guys are lucky. This is hard. Anyway, autosomal recessive diseases next. So we'll go into autosomal recessive diseases. You need a mutation in both alleles to express this condition. Example, 2 lowercase a's or 2 lowercase b's, not capitals. So recessive genes are shown by lowercase letters. For example, cystic fibrosis. This occurs because you'll have no functioning protein leading to the condition. So carriers are not affected as 50% of the protein is normal, and this is enough to get by. But usually one generation is affected when two parent carriers come together. So that's important to know, that when two parent carriers together, it's normally that it's just one generation that's affected. Cool. Something that increases the likelihood of autosomal recessive disorder is consanguinity, and this is what we talked about earlier, which are familial marriages. There would be a quarter or 25% chance of having an affected child, but that really depends on if both are carriers, or if there's one carrier and there's one normal, or if there's, it depends basically, I think, but I'm not too sure. But anyway, let's stick with it. There would be a quarter or 25% chance of having an affected child. Yeah, that's correct. So if an affected parent is having a child, actually no, it does depend on. For example, if an affected parent is having a child with someone who's normal, then all the offspring would be carriers, I believe. But anyway, I've messed this part up. Wow, shockingly. There would be, so anyway, let's stick with it. There would be a quarter or 25% chance of having an affected child. And that, as we'll see that here in the Punnett square. So if basically, oh god, this is too complicated to explain, but basically, if there's two carrier parents that mate together and have children, mashallah, then there's 25% chance of having a normal child, there's 50% of having a carrier, and there's 25% chance of being affected. You can only have an affected child with this condition if a affected is mating with an affected, or an affected is mating with a carrier, or carrier is mating with a carrier. You will not get the disease, and the children will not get the disease, if a affected or a carrier is meeting with someone who's normal. So, for example, this is why it's really complicated. Like, I'm overcomplicating this massively. But basically, this is capital T, this is lower t. If this was two capital T's, then obviously if you to work it out, this would be capital T, this would be capital T, lower t, and this would be capital T, lower t as well. That means there'd be 50% chance of having a carrier, and there'd be 50% chance of being normal, and you would not get affected child. And you can work out the Punnett squares, are very easy. And I'm really sorry for overcomplicating that, because that was just a mind melt. Anyway, characteristics of inheritance patterns for autosomal recessive conditions. So males and females are equally likely to be affected. The trait is often found in clusters of siblings, but not in their parents or siblings' offsprings. That's really important. The trait is often found in clusters of siblings, and it's normally in one generation. A rarer trait in the general population. If it's a rarer trait in the general population, then it's a greater chance of it being a consanguineous mating. So the rarer a trait in the general population, the greater the chance it was a consanguineous mating. All offspring of an affected person are obligate carriers. And that's something that I was trying to say when I had that moment of, I don't know, a moment of like proper mind melt a few moments ago, that all offspring of an affected person are obligate carriers. So if this affected person is mating with anyone, then they're either going to be carriers or they're going to be affected. If an affected person is mating with anyone, they're going to be a carrier or they're going to be affected. Cool. So here is a pedigree for an autosomal recessive condition. So the parents are not affected, which clearly shows it's an autosomal recessive condition. So that's what the first thing we're looking for. Are the parents affected? If they're not, then we know it's an autosomal recessive condition, like immediately. However, even if the parents can still be affected, and it still can be an autosomal recessive condition. Let's not get that wrong. But it's important to know that if the parents are not affected, then it clearly shows it's an autosomal recessive condition. But the parent can be affected, and children can also be affected. Nevertheless, the trait is found in clusters of siblings, but not in their parents or in their siblings' offspring, and that's generally what happens. Okay. So we'll go on to an X-linked condition. Anyway, so X-linked condition. Women have 2 copies of the X chromosome, whereas men have an X and Y chromosome. Therefore, if there's a genetic condition caused by alterations of X chromosome, it is an X-linked condition. And if it's recessive, it's more likely to affect men. It's more likely to affect men, as men only have 1 X chromosome. I'm going to repeat that again. Therefore, if there is a genetic condition caused by alterations in the X chromosome, it is an X-linked condition. And if it's recessive, it's more likely to affect men, as men only have 1 X chromosome. Men only have 1 X chromosome, sorry. So X-linked inheritance can be recessive and can be dominant. So when it's recessive, if it's recessive, it's where women are carriers, but males are affected. There is no male-to-male transmission, as man cannot pass on his X chromosome onto his son. If his child were male, he would pass it on his Y chromosome. However, there may be potentially male-to-female transmission, that in the sense that the father may pass on an affected or a dysfunctional X chromosome onto his daughter, and that could potentially happen, I believe. Anyway, going on to dominant. So X-linked inheritance can also be dominant. It's where women are also affected, but males are more severely affected and can be lethal. So if we were to look at the Punnett square for an X-linked condition, so here we would see the green indicating the normal gene or the normal allele, the red indicating the faulty allele. So here is our mother. The mother is the carrier of the X-linked condition. Here's the father. The father is normal. So if they were to both, if they were to have a child, then as we can see, the probability that they would either have a normal female or a normal male, or they would have a carrier female or an affected male. Cool? Cool. And really, if an exam, just draw out the Punnett squares, inshallah, it should be fine. Anyway, the characteristics of inheritance patterns for X-linked diseases. X-linked genes never pass from father to son, as father passes on his Y chromosome onto the son. Therefore, males are never carriers. Males are never carriers. Males are much more likely to be affected because they only have 1 copy of the gene. So if that gene is faulty, then they're affected. So males are much more likely to be affected because they only have 1 copy of the they only have 1 copy of the gene, as they only have 1 X chromosome. So if that gene is affected and it's faulty, then they're affected. Affected males get the disease from their mothers or are de novo or / new mutations. So affected males get the disease from their mothers, or these diseases are de novo or new mutations. All daughters of affected males are obligate carriers. So all daughters of affected males are obligate carriers. And probably the best tip and advice I can say is to do a Punnett square in the exam. So here's our mother, she is normal. Here's the father, he is affected. All the daughters are obligate carriers. They have to carry the disease because they will take the X chromosome from their father, and they have to be the disease, and they have to carry the disease. Allahu Alam, God knows if that means that they will be affected by the disease, that they probably won't be. Here we're saying that they are definitely carriers of the disease if the father is affected by the disease. Anyway, children of carrier female have 50% chance of receiving the mutant allele. So children of the carrier female have 50% chance of receiving the mutant allele. Here is our X-linked pedigree. As we can see, only boys are affected on the pedigree. Okay, that's something important. But that doesn't mean that females can't be affected, and we're going to come to this in a sec. So there are aspects of X-linked recessive inheritance that we need to talk about. Firstly, skewed X inactivation. Some women can manifest in the X-linked condition due to a skewed X-linked inactivation. So this means that normally inactivation of most genes on one X chromosome is random. It's completely random. But if inactivation of one X chromosome is favored over the other, if inactivation of one X chromosome is favored over the other, it will lead to an uneven number of cells with the same X chromosome inactivated and the other X chromosome activated. Therefore, this may cause the mutant gene to be activated more, causing a manifestation in the symptoms. For example, cardiac myopathy. So manifesting carriers are some women that do have some symptoms in X-linked recessive conditions, e.g. cardiomyopathy in DMD, which is Duchenne muscular dystrophy. There's also germline mosaicism, which we talked about before, which may be substantial in DMD or BMD, which are both types of muscular dystrophy. Okay, and moving on to the last part about mitochondrial inheritance. So mitochondrial genome is separate to the rest of the genome. It is inside the mitochondria. It's a small circular molecule that contains 38 genes. It is exclusively maternally inherited. It's exclusively maternally inherited. So homo is exclusively maternally inherited. Forgive me if I didn't stress that enough. It's maternally inherited from the mother. Homoplasy. It refers to the cells that have one type of mitochondrial DNA. Homoplasy have one type of mitochondrial DNA, which is either completely normal or completely mutant. Heteroplasia, on the other hand, they are cells that have more than one type of mitochondrial DNA. Well, some mitochondrial DNA are mutant and some are normal. And I'm going to try and get you a picture of this. Inshallah. Let's see. Yeah, so here's heteroplasia, where there's two types of mitochondrial DNA in the cells. As we can see, on the left there's some normal mitochondria and there's some abnormal mitochondria. If the percentage of abnormal mitochondria is greater, then the child may have a severe disease. If it's 50-50, then the child might have a mild disease. And if it's 20% mutant mitochondria, then the child should have no disease. So we'll go into further information about heteroplasia. So mitochondria sort randomly among daughter cells, and therefore they are cells where heteroplasia is present. So mitochondria sort randomly among daughter cells, therefore they are cells where heteroplasia is present. Each daughter cell may receive different proportions of mitochondria carrying normal and mutant DNA. Each daughter cell may receive different proportions of mitochondria carrying normal and mutant DNA. As we just saw in the picture, there may be different percentages carrying normal and mutant DNA. So even in our cells right now, we may have mutant mitochondrial DNA. We'll then go on to something called multifactorial inheritance. So this can be interaction between genes and environment. Some conditions are 100% genetic, such as DMD, as we said before, Duchenne muscular dystrophy, I think it's called. Others are 50-50, like ischemic heart disease, or spina bifida. And some diseases are completely environmental, like scurvy. An example is BRCA1 gene, which may lead to a person having breast cancer, colon cancer, or still not get cancer. So this is because some of the interactions which with the environment, which are important for BRCA1 gene to cause cancer. We then go on to the pedigrees, which are important. So I don't know why this is here at the end, it's a bit random, but basically pedigrees are important for three things: setting the scene, making a diagnosis, and risk calculations. Thank you for joining me. I'm sorry for that was not the best lesson. Jazakallah khairan.